A very good morning to one and all. With the blessings of the Almighty and our beloved founder, Chancellor, Colonel Dr. J.P.R. Sir, we've all gathered here virtually for celebrating the World Water Day, organized by the Center for Ocean Research, jointly with the Center for Remote Sensing and Geoinformatics and Department of Civil Engineering in association with Earth Science and Technology Cell, Ministry of Earth Sciences, and with the IUCN Commission on Ecosystem Management. On behalf of the Center for Ocean Research, I take a moment to express my heartfelt gratitude to our patrons and the higher management, Sathyabama Institute of Science and Technology, for their constant guidance, support, and encouragement in all our activities. Now let us get into the technical session. Today, we have with us Dr. R. Selva Kumar, Associate Professor in Nanobiotechnology, Head, Department of Biotechnology and Nanobiotechnology, PSG Institute of Advanced Studies. Before getting into the technical session, I would like to introduce our speaker today. Dr. R. Selva Kumar is an Associate Professor and Head in Nanobiotechnology at PSG Institute of Advanced Studies, Coimbatore, India. He earned his PhD in biotechnology from Bharathiar University, Coimbatore. His areas of research interest are application of microbial nanobiotechnology and nanotechnology for water purification, biosensors, and tissue engineering. He is a recipient of various prestigious awards like Water Advanced Research and Innovation Fellowship 2017 from Indo-US Science and Technology Forum, End of Year Research Fellowship Award 2015, awarded by Department of Education, Government of Australia. Bloston Postdoctoral Fellowship Award at Zuckerberg Institute for Water Research by Government of Israel 2013. Invention Award on Fabrication of Three-Dimensional Scaffolds for Tissue Engineering from Intellectual Ventures, USA 2012. Apart from that, he's also been honored with CSIR SRF, DRDO JRF, SRF, DST International Travel Grant, etc. He represented India as a guest speaker during the India-Brazil South Africa Dialogue Forum on water treatment and sensors for water pollution detection held on 4th and 5th April 2014 at Birchwood Hotel and OR Tambo Conference Center, Johannesburg, South Africa. He has received research projects for a total value of 4 crore from various funding agencies like IGCAR, DSC, DRDO, BRNS, ONGC, TNSC, ST, ICMR, and Wilbro, etc. He has authored 86 international and national papers and 6 book chapters with a H index of 23 and I index of 49. His research articles have been cited more than 1,700 times by various researchers around the world. He has supervised 2 NPDFs, 4 PhD, and 4 MPhil students so far. Currently, he is guiding 5 PhD students. He has 2 Indian patent fields and 3 technology transfers to his credit. He is an active reviewer in various international journals like Water Research Journal of Hazardous Materials, Chemical Engineering Journal, International Journal of Environment and Waste Management, Journal of Colloids and Interfaces B, Waste and Biomass Valorization, Separation Science and Technology, etc. We are pleased to have you with us today, sir. Over to you, sir.
Thank you, Bhuvaneshwari. And I thank Bhuvaneshwari especially for giving a nice introduction. And probably this is the first time someone has not skipped anything. And she managed within a very short duration of time. Thank you for that, Bhuvaneshwari. It was really nice to hear from you. So first of all, I thank Inba, my friend, and for giving this opportunity, and especially on a great occasion like Water Day. So where my bread and butter is water. In fact, so I should say it's a solid liquid which is different.
Actually, I have to thank you because the entire program I framed only because of your help. Actually, you only decided who are the speakers, and you only helped me to identify the speakers, and even our Vice Chancellor, she appreciated about the speakers and their topics. So first of all, I have to thank you for this opportunity and you are being here with us today. Thank you.
Yeah, you are good. So, okay. I know that Inba is a very humble person, so I don't want to more comment on that.
Okay, thank you.
So, anyhow, it's great working with you, Inba, and with the Sathyabama team, basically. And I also thank the university for giving me an opportunity to be with you all on this great day. In fact, one day after the great day. So today I will move to my lecture. It's on emerging contaminants. Okay. Okay. I hope you will hear the disturbance now from the background. I'll try to move my mic a little bit closer so that you will hear my voice. Emerging contaminants, as Dr. Paramita talked about plastics yesterday, it's not only the plastics which is an emerging contaminants, there is a whole lot of list where they have classified many chemicals as an emerging contaminants which are not currently under the scrutiny by the boards. Okay. These are emerging and they are getting diversified daily, and the byproducts of some chemicals are when they undergo degradation in environment and as well as into the in the other sectors, other industrial applications also, they are also classified as emerging contaminants. So basically, there is no norm so far for these chemicals when they are found in the water. So, for example, if it is arsenic, you have very ppb level of minimum permissible limit. So, like that, there is no such norms for emerging contaminants in water and environmental samples. The biggest challenge of emerging contaminant is that the protocols for the quantification, which is the most important thing. Although we have some methods like Gcms, like what Paramita was talking about yesterday, we have some protocols, but however, the extraction of EC for the analysis is a very, very challenging field. Okay, because we are not sure whether we have extracted clearly, since we don't have an optimized, or what to say, like a SOP, standard operating protocol. That is not there. So what is the challenge what we are going to do, what we are going to face when you are really trying to extract the emerging contaminants in water for analytical purpose? So that is what we are going to discuss today. So basically, the emerging contaminant, or it's also called as contaminants of emerging concern. It was renamed by US EPA in 2017. So emerging contaminant, in definition by US Geological Survey, states that any synthetic or naturally occurring chemical or any microorganism that is not commonly monitored in environment but has a potential to enter into environment and cause suspected adverse ecological or human health effects. So the definition is very clear, and it states that, okay, it has not been monitored so far. So that is what it really states. Okay, so it's not new. Emerging contaminants has been a focus recently, but said that it is not new to the environment. It has been already there, but we were not monitoring it earlier. But due to the advent of the analytical advances, now we have the capacity to detect them at least. Okay, so and lot of research is needed to optimize them and bring a possible what to say the level, acceptable level in the environment. So that has to be decided based on the analytical protocols. Okay, so that is one thing, and it already existed, and we are recently identifying them. Okay, so EC are generally classified into several classes, like based on the origin and function, application, their action mechanisms, and some are yet to be classified. Okay, but they are classified into small or specific classes, not into the main group. So this is still a developing field, and proper classification is yet to come for a EC. Okay, so how the bibliometric analysis clearly indicates that people have started showing interest in the field of emerging contaminants. So the number of publications every year is increasing, and if you can see here, earlier in 2000, there are very few numbers, like even 10 and 15 publications were there. But after 2010, especially, there was a huge spike in the number of publications. Okay, and the countries who are involved in the research of emerging contaminants also increased. in parallel with the publications. Okay, so in 2018, almost 1,000 paper per year was achieved, and I think it is still growing in recent years. And so a lot of people, a lot of countries have started working on it. Okay. Now, if you see the area in which the publications are made with respect to emerging contaminants, you can see that environmental science, as usual, as expected, it is the maximum area where EC has been researched, and then it followed by chemistry, chemical engineering, and agricultural, biological sciences, medicine, like that. Okay. Now, recently, emerging contaminant in medicine as well as this pharmacological studies is increasing. Very less studies are there in agricultural and biological sciences. Okay, so environmental science and chemistry are the leading aspects as expected, because these are all chemical compounds. So we expect these three areas to be dominating with respect to the publications. Okay. So where does India stand on this? If you see the 2005-2009 statistics, India was almost, we can say that it was among the 28% among the all other countries, and we didn't have real stand for the publication till 2009. However, after 2010, or sorry, 15, we have a substantial place in terms of the EC research, and you can see here closely we have a good number of competing number of publications in the area of water research. Okay, sorry, environmental emerging contaminant research. Okay, so after 2010, we are one among the leading countries. Okay, you can see here, this is as per 2000 to 2019 data. India published more than 188 publications, and with a citation of 2507, and the citation per paper was 13.3. Okay, so closely we have started doing a lot of research, and India is leading now in terms of emerging contaminant research. So how do you classify the emerging contaminants which are entering the environment? It is very diverse, because every chemical which we use in every field is a potential emerging contaminant, and because we are not actually analyzing the fate of the environment, emerging contaminant EC, and the most prominent fields which are where you are directly using the chemicals which are a potential EC are pharmaceutical industries and veterinary products, disinfectants and biocides, drugs, personal care products, industrial chemicals, food additives, even water disinfectant byproducts, nanomaterials, where we work extensively, is a emerging contaminant. Pathogens which are not under classification are also EC, and biological toxins. So you can see that the deviation of emerging contaminant starts from a biological molecule to a chemical molecule. So it's very diverse, and the most common ones are like PFBs and PFCs, perfluorinated compounds, brominated compounds, flame retardants. All these things are potential ones. Okay, just a minute. Let me hold this here. Okay, so it's all a cycle how it reaches human beings, plants, and microorganisms, and we will see in detail about those things in the next slide. And which area in EC is highly researched? Okay, now we are told that EC is India has started doing a lot of work. If you see this data which was published in 2020, it clearly states that almost all the countries, starting from USA to India, all are almost following the same type of research, and the similar components have been looked by all the countries. For example, if you see here, China looks more on China, and Germany looks more onto microplastics. But said that other countries are also equally working. Only few small deviation is there in the percentage. Lot on pharmaceutical, personal care products, and organohalogens, and heavy metals, steroids, and hormones. If you see Indian concept and Indian researchers' mind, if you see that, you can see that heavy metals have been potentially looked into when compared to when compared to other chemicals, okay, and pharmaceutical compounds. So these two, because India being the pharmaceutical hub for the world, more focus is on the pharmaceutical and personal care products, okay, and followed by that we have heavy metals. So India also is following the same pattern as that of the other industrially viable nations, okay. So if you see the concentration of emerging contaminants in the ecosystem, every ecosystem differs, and these are some of the reports which has been published from throughout the world, okay. For example, if you see here, urban wastewater in China has more of bisphenol and salicylic acid, and still in nanogram level, okay. And commercial seafoods of Europe has more of halogenated flame retardants. So you can see here the seafood which they are exporting has a flame retardant in it, which we are eating. Okay, so the concentration, especially here, 41.1 nanogram per gram. So still in nanograms, right? Then sewage, groundwater, river water. This is a big pool. And in Tokyo, they reported that nitrosodimethylamine is there at a range of 10 nanogram to 5 mg per liter based on these ones. Okay, it may be within this zone. So drinking water in Germany has PFOA, and USA has 1,4-dioxane, and you can see the concentration here is huge. So this clearly states that every country is now facing the problem of emerging contaminants in water, food, as well as soil. Okay, so all the sectors are being contaminated by emerging contaminants, and the concentration of EC is still in nano, except in few cases where nanogram, except in few cases where they have reported in mg per liter. So I really want to thank Matthew and Convenor. They are from Anna University, and they have made a very good. Although the journal is not so good, but the compilation what they have did is very good. Okay, so they have stated how India is placed and how researchers in India is they have worked on the emerging contaminants, especially in Indian waters and rivers. Okay, so if you can see here, Kaveri, Tamirabarani, Vellar, and many other places, Varanasi, Ramnagar, all these areas have rivers, perennial rivers, and these rivers have enough of EC. Okay, so you can see the concentration all in micrograms. So few are still in nanograms, of course, and you can see here this antifungal compound which is in Hyderabad. You can see the concentration, how much they are. So pretty diverse, and you starting from a nanogram level to mg and what to say, even grams. So that is a big challenge for us to treat, because we don't have a system directly in our ETP or in the purification plants which can handle these pharma compounds. Very few compounds have been reported to be absorbed by the existing treatment system. But on the large, we don't have an essential effective removal system for this emerging contaminants. And why we have to bother about this? Because this is going to directly impact the health, and you know that the rate of cancer increase in India is very high. So therefore, it is essential to understand what actually they are actually doing. Okay. Yeah. All right. Is the slide moving? Hello.
Yes, sir. Yes, sir.
Yeah. Okay. Okay. Fine. Good. So next is pesticides. You can also see the concentration all in micrograms, except few like chloropyrifos in nanogram. So all these things are sorry. So and it has been reported throughout India, MP, UP, and Assam. All these areas have been reporting. Their researchers have started reporting that yes, AEC is worth looking into, and we have to work on it. Okay, personal care products like triclosan, UV9, UV329, DEET, all these things are there in water, and again they are also present in most of the river bed. So water contamination with uh, emerging contaminants is a bigger concern than what we are thinking. Okay, and we have been focusing only on arsenic, fluoride, nitrate, and few other compounds. Recently, few radioactive compounds have been brought into preference. But apart from that, these chemicals are the real culprits, which has to be looked into. Okay, so we have to really focus a lot on various aspects of handling this emerging contaminants. Okay, so how does this emerging contaminants actually reach water? So basically they all reach water from a different sources. So for example, in agriculture field, we are using pesticides. The crop pest application is there. Then from that crop, it reaches the soil zone, and through leaching, it reaches the unsaturated zone and then to the groundwater. And of course, in between, surface water is also there. So therefore, the deposition onto the surface water as well as groundwater is likely. Okay, and landfill and leachate is another area where it's especially where Dr. Paromita was talking about the plastic leaching from the waste dump yards. So that is a bigger challenge because they have this leak because they are open ground, and any rainfall, heavy rainfall or any environmental climatic condition is going to influence the leaching of the EC from the dump yards into the groundwater. Okay, so that is again a major challenge. And then pharmaceutical and personal care products directly reaches us through the domestic and hospital waste effluents and through septic tanks to the groundwater. Okay, now if the system doesn't, and most of our existing sewage system does not have any of the EC based removal system. So therefore we need to develop new materials, new concepts to remove this EC from the water before they are sent back to the environment. Okay, so industrial compounds. This is although this is only one box. Most of these other boxes are controlled by the industrial compounds. So basically, the industry producers produce all these compounds. So industrial compounds directly, apart from the consumer part, they are also contributing directly to the surface water. Till now, we don't have a norms on how much of the emerging contaminants are released from the various industries like heavy metal, so like electroplating industry or pharma industry. We don't really have a bigger picture on how much it is being released into the water sources. So after that, we are taking it, and we are taking the groundwater for drinking. So ultimately, all these chemicals end up into your body, and the consumer is actually affected by this. So you can see that the stronger arrows are pathways, and minor arrows, the dashed lines are the minor pathways in this. Okay, and all the shaded ones are receptors. So basically, when either before a water reaches a consumer, we have to make sure that the groundwater and surface water are treated effectively, that for EC, and it has to be tested, and qualitative as well as quantitative data is required before it reaches the consumer. Okay, so we have to really focus on these pathways. Okay, so what is the potency of this emerging contaminants, and which compounds reaches much faster into the environment? So the distribution of common EC, that is very much important, and this is based on two factors like polarity of the compound and the volatility of the compound. Okay, so if it is going to be non-volatile, mostly it takes more time to reach. If it is volatile, then it automatically goes to the environment into the air, basically from water. So volatility and the lipophilicity and hydrophilicity decide the distribution of the EC. Okay, so if someone really wants to to start start a research on EC, they have to first look into the polarity and the volatility of the EC, and then decide, okay, now what is going to be the impact. Okay, and you can see here all chelating agents, X-ray contrast medias, metabolites, pesticides, phenols, all have their own level of volatility when you draw a graph against polarity to volatility. Okay, so therefore the selection of EC has to be done systematically. Sorry. One more important thing on EC bioaccumulation is on the lipophilicity. Now, if a compound is having a higher lipophilicity, okay, then its bioaccumulation into the environment is going to be very high. Okay, so because it goes directly into the fat molecules into the body system. Okay, they get deposited in those things. So when an animal eats, for example, a deer eats a leaf which is having a pesticide in it, so it directly goes and gets deposited, and it induces further the clinical issues. Okay, now therefore lipophilicity and hydrophilicity has to be parallelly looked into along with volatility and non-volatility of the compound. Okay, so basically you have to understand the EC from this perspective, and where does this stand and what is going to be the impact, that we have to understand clearly. Okay, so said that, so what is the challenge or what we are going to face? First is the wide range of compounds. It's not a single compound. Like when we talk about arsenic, we either talk about AS3 or AS5. It's not like this. The same compound which can break into hundreds of compounds. Okay, the mother compound can break into hundreds of compounds, and each compound or the byproduct of the compound itself is a EC, which is either not detected or which has been, we don't have a system to be detected. Okay, so these two, the wide range of compounds is one of the major issue. Then the concentration of the compound is again an issue. So mostly these ECs are in picogram to nanogram level, where we underestimate that, okay, the picogram level or nanogram level in the water is not going to have a bigger impact. So that is a misunderstanding basically. So even a lower concentration of a product and its continuous accumulation into the body system is going to cause health effects. Okay, so therefore we have to understand what is the concentration it is there and its physical chemical properties. Okay, all these three things are related. So wide range of compounds with specific physiochemical properties at lower concentration. That is the biggest challenge. Okay, the second is source type and the route of entry and migration identification. So we, although we know a pathway is a larger pathway basically, if you go into the individual pathways of the same pathway, then it's going to be very massive. The research what we need is going to be massive, because we have to first understand the interaction of the EC with the source and its conversion or its fate in the source, and then we have to go to the type, what type of EC is going out of the source and how it is entering into the different environmental systems. So, and whether it gets stopped in one environmental system or it reaches the consumer at the end. So all these things needs a systematic study, which is really time taking, and as well as a lot of money is involved on the research part. So therefore the source type and route identification is a very, very important one. And then the persistence, how long the EC stays in the environment, that is going to be important. So as I told you, the mobility of this EC or the EPs is going to be decided by polarity, volatility, persistence, and many other physiochemical properties of the EC. So all these things are going to be playing a major role when you start researching about EC. Okay, so mobility basically depends on the property of the EC and its the byproducts which are formed. Okay, so bioaccumulation and microbial degradation, again, few of the compounds are readily taken up by microorganisms. So therefore microbial degradation is possible. Now the next question comes here is that whether the microbial degraded products are safe or not. So it may be potentially, Pseudomonas putida, we were talking about oil degradation. Now oil is degraded, right? Fine. So what happened to the byproducts after the degradation? Whether they have to be removed or not, or whether how potentially they are dangerous to the environment. All those things needs a lot of research, and how we can remove it using an adsorption matrix. So development of adsorption matrix has not been done widely now. So people are working on that, and that is itself again a challenge. And we don't have a regulation for EC now. People US EPA as well as other regulating bodies are coming up with norms. Okay, they have given a list called as contaminant candidate list every year, and this CCL changes every year. So we have to keep updating the CCL, because every here and there people are finding new contaminants in water. So therefore this contaminant candidate list has to be upgraded every year, and a new monitoring system and regulation has to be framed for that. So it is not that only one time people come to the table and list a regulation, but it has to be done on a continuous basis. Okay. The biggest challenge, the next biggest challenge is the analytical methods and instrumentation. So far, all these ECs are mostly identified and quantified based on very high-ended costly equipments, which many of the researchers are not able to afford. And few who have a very good funding and infrastructure facilities, they do have the capacity to develop methods for identifying EC. So therefore, we should encourage higher, the institutes to develop further infrastructural instrumentation to handle ECs. Okay, and very limited scientific publication in this area. Okay. All right. Yeah, 1 minute. I'll take this down. Okay. So I don't know how to put this down. One minute, just a minute. Just want to put this down. Okay, let it be there. So the interaction between the pollutants is a major impact. Okay, so this is the challenge of emerging contaminant. So how to understand the fate? So far what we have understood is the complexities of the emerging contaminants in water, and what is the fate of these contaminants. So let us take somewhere from here, emerging pollutants. Okay, so can someone help me on this? I really don't want to see this. Okay, so the emerging pollutants basically they are reaching all the 3 layers of the environment, like air, soil, and water. So emerging contaminants, it gets volatilized and then it reaches air, and then through rain it precipitates back and it goes down into the water. So same case happens here. They get there is a bioaccumulation factor and degradation factor. They get ingested and it can reach the vegetation through the soil as well as the water. And the sunlight itself can degrade them, and new products are formed during the dry deposition process, and it gets degraded and new ECs are formed. So therefore, this is how it gets spread. Okay, and in case of the volatile compounds, there is a diffusive exchange between the soil and the air, which also is carried down to water. Okay, so all these things are very much important, and so in order to understand the fate of EC, we have to have a complete knowledge on the chemical structure of EC, followed by physiochemical properties and the combination effects. That means what happens to the chemical structure when it reaches the water or the soil or in the air. Okay, so whether this chemical structure remains the same, and if there is a change, what is the physiochemical properties of that changed molecules, and how the new molecule is going to interact with the environment, whether the parent molecule interaction is the same with the byproducts. So that is again going to decide the fate of the target disease on bioaccumulation and entry into the every system. Okay. So obviously, when we are talking about the accumulation in biomolecules, toxicity is going to be there. So mostly in wastewater, what is the problem? The toxicity is because we don't completely remove it, or we don't remove it at all. And then the EP enters rivers and drinking water system, surface runoff and agriculture runoff. So all these things are going to give an accumulation, and they are going to be toxic. So these are the sources by which these molecules get entered into this one and becomes toxic to the environment. Okay. So far, Inba, how much time I have? Hello.
Selva, you can go ahead. So no problem.
Okay. So how to select sample matrices? So what are the considerations scientists should have, or the researchers should have when they are selecting a sample matrix for EC? So first, understand the physical chemical properties, as I have told earlier: volatility, partition coefficient, and the properties of the interacting medium. So that means if you are talking about a soil, you should understand the properties of the soil and the composition of the soil, and how likely the EC is going to interact with this. Whether the EC is going to be permanently attached to the soil matrix, whether there is a strong adsorption between the EC and the soil matrix, or it's going to be a weak one by which the mobility is going to be increased. So that has to be understood. So not only the EC property, but also the interacting media property has to be clearly understood when we deal about the EC. Okay. Then the hydrophilic compounds in water. So, for example, if the water is already contaminated with few metals, the EC can interact with those metals, and then they can form a complex. So there is a complexation possibility of EC with other contaminants present in the water, okay, or other chemicals which you are adding into the water. So that has to be considered. Okay, so you should analyze the possible contaminants or the co-chemicals which are present in the interacting media. Okay, that has to be there. Okay, and the hydrophobic compounds in suspended matters, sediments, and biological tissues. We call it as log KOW, which has to be greater than 4. Okay, so if it is going to be there, then your likely accumulation into the matrix is going to be high. So that has to be done, and then selection of a proper analytical method, that is very, very important. Okay, and for example, if you take PAH, polycyclic aromatic hydrocarbons, then GC is the best preferred method, and it is more represented in sediment sample than in water. Okay, so GCMS gives you a clear-cut idea on how much PAH is there. Okay, and how much sample you have to have. That is very, very important. You cannot just take a few micrograms of sample and represent an entire interacting media. So therefore, you should have enough quantity. I said why we have to have enough sample is that, since as you saw in the literature which I presented, most of the concentration of EC is either in picograms or in nanograms, except few which are in micrograms and grams. Okay, so excluding those things, others are already in a very nanogram or picogram level. Therefore, what we have to consider is that we have to have enough sample to meet the sensitivity of the instrument. Okay, why? Because there should be some detection limit for every instrument and for every method. So instruments, we have to match the sensitivity of the instrument by having more samples, preferably a concentrated sample for the analysis. Okay, so therefore you have to extract the EC from the matrix, concentrate it, and then you have to go for a analysis using appropriate instrumentation. So that has to be kept in mind. Okay, so contaminant sources fluctuate because of the environment factor. So you have to have a appropriate periodic representation of sample. Okay, that means you should have samples from during the monsoon period, pre-monsoon period, or post-monsoon period. You should have data of the same water or the same ecosystem at different climatic intervals. Okay, so therefore you will understand what is the fluctuation of the compound. Okay, so based on that, you can probably select the sampling time. Okay, so that is very important for EC. Okay, so probably you have to select the sampling time from that. Okay, and sampling method. Okay. Water samples mostly done by passive sampling methods are common in evaluating ECs. Okay. For example, such as passive sampling by collecting medias, which are made by man-made devices. So we also have to have a time integrated average concentration of organic contaminants. All these things are in water samples. So that's why I was talking about the climatic condition, the duration and during which we collect the samples, and how much concentration is there on an average during a period. Okay, during a year or during a considered period. Okay, then sediments and biological samples, they have to be why we have to go for sediments and biological samples. Just in the same, for example, if you are working on a river EC contamination, just collecting water sample is not sufficient, because it's a flowing water, and therefore it may not be a correct representation of this one. However, there are the soil or the sediments which are present in the river, which may be a potential source to understand how long the contaminant is there in the system. Okay, so therefore, when you are working on a water sample, just don't focus only on the water sample, but also the sediments and biological samples which are collected from the water sample. That is going to give both the short-term as well as long-term data on the prevalence of EC in the environment. Okay. In case of air samples, of course, you have to have graph sampling as well as the continuous sampling. And then what you have to do is you have to identify what are the possible semi-volatile and volatile compounds present in the air. And then you have to basically, wherever you are handling air samples, you should have instruments with head spacer where we can handle volatile compounds. Okay, so what are the sampling techniques? If you are going to use water sample, graph sampling is less preferred. Passive sampling and event-based sampling is more preferred. And in case of sediment sampling, you should have passive samples or graph samples. For biota sampling, graph samples, event-based samples. Air sampling, you should have the appropriate filters to grab the ECs from this. Okay, so these are the techniques which are generally used for sampling ECs. Okay, this is the most important section of the EC. How you will prepare a sample. Okay, the sample preparation for EC depends on the type of sample. That is clear, that everyone knows. But how you are going to extract it from that particular domain is real. It needs a real expertise. Okay, you should understand the EC which you are targeting. Okay, basically screening is done, and you should prioritize which compound you are going to test, and based on that you have to devise your sample preparation method. Okay, and you should understand, based on the screening you will already know how much quantity of the EC is going to be there and what are the expected byproducts. So all these things are very much important to prepare a sample. Okay, to maximize the sample amount of sample analyzed, you have to minimize the interference. Okay, so interference are going to have a bigger say in EC than in case of metal ions. Why? Because the interfering compounds here are also in the nanogram, and your ECs are also going to be in nanogram, and there is a huge potential to form complexes between the interfering ions and this one at nanogram level. Okay, so therefore you have to minimize the level of interference in this. Therefore, you need pre-extraction protocols, okay, to reduce the interference. So that's how we are going to concentrate the EC in the sample. And then after concentrating, you have to go for a cleanup step. We will discuss about both the prior extraction and the cleanup steps in future. Okay, in the forthcoming slides. The extraction method from environmental matrices. For water sample, you have to use liquid-liquid extraction using non-polar and non-water miscible solvents, and then solid phase extraction is also being used. In case of sediments and soils, you have to use solvent, microwave assisted. There are a lot of methods for sediments and soil. These are only few which I have listed. Okay, microwave assisted, sonication assisted, bead beater, and there is a new method called as this future. Which is quick, easy, cheap, effective, rugged, and safe. That's how they have named it. Yeah. Sorry. Hello. Yeah, thank you. So then in case of biological sample, you have to use either a pressure extraction or bead beater and freeze thawing are the methods. And in case of air, you have to have oxalate extraction and passive sampler and solid liquid extraction. All these things are the extraction methods used for water samples. Okay. Huge list of methods are there for easy from aqueous and solid matrices. I don't want to read everything. Instead, I will go into the each methods. So first, most common is the liquid-liquid extraction method and continuous liquid-liquid extraction method. These two are more common.
Rajendra.
Yeah. Thank you.
Vidit,
So,
if he is again doing that, please remove them from the meeting.
Yes, sir.
Yeah, thank you. Thank you, Inba. So the next one is the appropriate selection of the solvent system. Mostly pentathione and pentanehexane, cyclohexane are used, and they are used to partition the compound into the from the soil and water matrix. Okay, so basically this method is quite commonly used. Okay, but the problem here is the volume. So what the quantity of the solvents what you add to extract the lower doses is huge. So therefore there is a need for reducing the volume of sample and after which you can concentrate. Basically you have to, after extraction into this, you have to reduce the volume. Okay, and then derivatize it according to the analytical instrument. So you should know what is the capacity of the analytical instrument, and then you have to derivatize it if it is needed. Okay, as an alternative for the volume of solvents which are being used for extraction, continuous LLE is used, where you use less solvent and extraction time is also less. The next method is microwave assisted extraction. They use non-ionizing radiations, and the volume of sample required is less and time is also less. Okay, but you need the appropriate instrumentation facility for doing this. Okay, multiple samples can be extracted at the same time. Yes, obviously you can. What we do is we extract. We use this method for extracting closely to 12 samples at a time. Okay, so multiple samples we can use it either as an open method and closed vessels. We use closed vessels here, and you can irradiate it and then extract it using the solvents. Okay. The next method is called as DLLME, which is dispersive liquid-liquid microextraction. All are the same type of solvent extraction steps, but few modifications are there. Okay. So if you see here, the sample is taken and then the organic solvent or the dispersing solvent is actually injected rapidly into the sample. Therefore, this rapid injection, what it happens is it travels throughout the samples and collects all the EC more effectively than just slowly adding it. Okay, so injection with a force, injecting the solvent with a force inside the matrix basically improves the extractability of the solvent, extractability of the EC from the water sample. Okay, then what you do, you vortex it and centrifuge, and then you again give the second solvent. First solvent is the extracting solvent, and second solvent is called as dispersing solvent. Okay, so you give a dispersing solvent, and then the collected samples are analyzed in the Gcms. So the next method is a vortex method. Again, I told you this is again a small modification of the existing the organic liquid extraction process. So VALME is called as vortex assisted liquid microextraction. Basically, instead of an injection module, you are using a vortex model for the extraction of the EC. Okay, so this is widely used for micropollutants in many matrices used to detect chemicals, specifically micropollutants. Okay. The next widely used is single rope micro extraction. All this, you see, why I have listed all these things is that EC has to be fitted according to the the extraction protocol has to be fitted according to the EC which is there in the environment. Okay, so after screening, you should basically use these methods to extract and then concentrate. Okay. So three different methods are there with respect to single drop microextraction. One is direct immersion method, hollow fiber method, headspace method, and it all depends on how much money you have. So if you have more money, you can go for hollow fiber or headspace extraction protocol, and then you need an equipped lab for handling all these things. Okay. So basically, you drop a solvent on a sample, and the solvent travels into this in the presence of a magnetic stirrer, and then you collect it from the headspace and do it. Okay, so the same thing is happening. So different hollow fiber also does the same thing. Okay, you can see here it passes through the syringe, and then the samples are here, and the solvent basically picks up the EC from this from the donor phase, and then the acceptor phase. From the acceptor phase, you can pull it out. So by this way, you are able to concentrate the EC in this one without disturbing the sample matrix. So how you will do it in solid phase extraction? Basically, we need a different type of cartridges for this. And of course, solid phase extraction is more an what to say laborious process, because you have to redo the samples a lot of time to till you don't get a EC in it. Okay, so that much time you have to repeat the same process. So that is one of the challenge in solid phase extraction process. And of course, you are using cartridge matrices where the EC gets absorbed to it. Okay, so there are different formats of solid phase extraction protocol. One is the thin flat discs, cylindrical cartridges, and bell plates. And a lot of sorbents are used, silica based, hydrophilic, lipophilic, balanced ones, and microoxidation. All those things are there for the solid phase extraction process. This is one of the example of using solid phase extraction process. So what you do is you extract, basically take the liquid sample, and then you use a normal syringe and filters, and then pack the silica gel matrix here, and then you add the water sample, filter it, and then compress the cartridge, and then you add your sample, and you get the pesticide dissolved into the water, and then the pesticides move to the filter and remove the water, remove the filter from this where you have the pesticide, then extract it. Basically, it's a concentration process for the lower concentration ECs. Okay, so you can concentrate it like that, and then you can inject it into the space. So you can also use micro extraction model. This is again fiber withdrawal process. You have a fiber at the edge and the sample, and then you suck it up. So therefore, the sample passes through this, and there is a filter or the fiber here, which is taken out and put it into GC, and then from the GC the compounds are taken out. Okay. So different type of matrices have been developed based on the SPME. And you can see here thin films, micro extraction, packed sorbents, magnetic field sorbents, dispersive solid phase, in tube. Each one itself is a science, and therefore we don't have time to go into each one of these methods. Matrix solid phase dispersion is again a method. So basically, what you do is you add olive oil to the sample, and then blend it, and then put it into a column, and then compress it, and then elute it using a dissolvent. So then this is called a solid phase dispersion. Okay. Basically, this is a method which is used for solid samples, and then and this is preferred for LCMS type of analysis. So in case of semi-volatile or volatile compound extraction, you can go for these methods which we have already discussed: liquid extraction, solid phase, auto trace, or solid phase micro extraction. And if it is a solid sample, you have to go for Soxalate or accelerated solvent extraction process. Ultrasonication is widely used to disperse or to remove the EC from the solid matrix, and then clean it and then analyze it. Recently, nanosorbents are also used for the EC extraction, and you can use many matrices which we have described: nanofibers, metal oxide nanoparticles, silica-based nanoparticles, carbon, magnetic. So a lot of nanoparticle types are being evaluated for its absorption of EC, and of course, all these things can be put together with the technique, appropriate technique like the SPME what we were discussing in the previous slides. Okay, sorry, I have to rush it up considering the time. And after extraction, now you have got the EC into a solvent system and it's ready for injection. Don't go directly for the injection because there is a very high likeliness of co-contaminants in it. Therefore, you need to clean it. Okay. So, as I said, why we have to clean it? Because you have to remove the interfering compounds like lipids, peptides, and sulfur. And then, if you are going to use a GCMS as an analytical method, definitely your samples should be lipid-free as well as sulfur-free. Okay, to get very less interference in the data. Okay, and if you're going to use LCMS, they have to be free from large molecules like proteins, peptides, and lipids. Okay, why? Because there is going to be an ionic suppression of EC when these compounds are going to be there. So this is the requirement for GCMS and LCMS with respect to cleanup. How you have to clean it such that there is no lipid, there is no large molecules in it. And what methods you are likely to you are supposed to use is size exclusion chromatography, which cleans lipophilic macromolecules, gel filtration chromatography for hydrophilic molecules, liquid-liquid partitioning, or normal phase. Okay. So, what are the instruments widely used? So, these are sensitive methods which are used for detecting the EC in ppb or ppt level. And then most of the results what you see in literature, they have used either of the three methods like GCMS, GCMS-MS or MS-MS multiples, and LCMS, ICP-OES, all those things are used for the sample. Okay, if you are going for GCMS, make sure that your sample should be volatile and derivatized, okay, to ensure the volatility. Okay, that means if you are selecting GCMS, most of the compounds should be volatile enough. And LCMS also, then your sample should be hydrophilic in nature, polar or waterborne. Okay. And in case of ICP, mostly done for cationic samples like metals and metalloids. Okay, so this is how you have to process the sample for analysis. This we have covered. I told you biological samples, you have to go for solid extraction, solid sample extraction as well as liquid sample extraction, and then you have to select appropriate liquid extraction process, and then purify it, go for the purification, and then mass spectroscopy, and you can add TOF to it. That will give you more precision. Then organic compound detection is again you have to use mostly GC for this. Okay. Let's skip this. So these are some of the EPA methods which is available for the semi-volatile compounds. US EPA method 543, 525, 625, 8270, and it is especially for in case of the organic chemicals in drinking water, you have to use this one. Semi-volatile compounds, you have to use 525, and for municipal and industrial waters, you have to use 625, and semi-volatile organic compounds using GCMS, you have to go for 8270. Okay. So these are the latest analytical methods for different diseases. For example, if you are going to detect disinfection byproducts, then you have to use LCMS, GCMS, LCMS, and ion chromatography. For pharmaceutical compounds, you have to use high resolution mass spectroscopy or LCMS, and dioxide you have to use GCMS. So selection of the method and the analytical instrument is very much important. Okay, and these are some methods which are there. All these details you can find it in www.epa.gov if you are from literatures. Okay, now most of the removal methods are mostly multiple methods are brought together to remove EC effectively. Okay, and this is just a data which shows what are the compounds which are being researched more for the treatment of EC from water. Okay, so for example, the methodology wise, okay, so this is oxidation based methodology. So you are going to use either a ozone based methodology or ultrasonication, sulfate radicals, Fenton-based electrochemical oxidation, and photocatalysis. So if you see that most of the research is in the photocatalytic sector for oxidation. And in case of adsorption materials, as usual, as expected, carbon-based matrices are going to be very effective for this one. That means that a lot of research is on that line. Okay, and the effectivity wise, membrane wise, reverse osmosis membrane, nanofiltration, ultrafiltration membranes have been researched. So always we can use different hybrid methods to enhance the efficiency of removal of emerging contaminants from water. For example, activated sludge along with membrane separation and gamma radiation is used basically. For in case of constructed wetlands, coupled with waste stabilized ponds, you have to use adsorption process along with biodegradation, volatilization, and photodegradation. All these four has to be used. And for removal of pesticides and pharmaceutical compounds, you have to use MBR along with other filtration matrices. And for few other compounds, we can use UV oxidation process along with activated carbon. So basically, every compound has to be optimized with the permutation combination of the existing methods to see which one is going to be more effective. Okay, for endocrine disruptive chemicals, you can use flocculants, activated sludge, and ultrafiltration matrices. For antibiotics, you can use ultrafiltration, activated carbon, and others. So all these emerging contaminants require one or the other methodology which is commonly available for the treatment process, and therefore proper permutation combination has to be devised for a particular EC. Okay, this is a bigger subject which I will skip considering the time. These are some regulations which are present in WHO and US EPA, EU, and ISO. And these are the method numbers, okay, which is available for this one. This is all compiled by Matthew and Convenor, and you will find that. And just to share, this is one of the method which was one of the monitoring system which was developed in Germany. It is called as Norman decision tree for groundwater. Sorry for the spelling mistake. It's a groundwater monitoring of EC. So you have to find potential. What is the potential of the groundwater exposure of for a particular chemical that we have to analyze? And if the potential is going to be high, then you have to systematically go down to the step 2 and step 3. Okay, and then you have to propose whichever is going to have a higher biological significance. You have to start regulating them and monitoring them at different levels, and then you have to finalize which is a priority chemical, which is non-priority chemical. Okay, so how to find a solution? This is again, solutions is the methodology which they have developed, and how we are going to handle it. So it's not that we just identify it, it has to be monitored, and what likely is going to be added new to the environment is also to be determined. So we have to start with effect based and chemical monitoring and EDA and environmental disaster analysis, and then we have to analyze the exposure and risk modeling, and then the future scenarios. Based on this, we have to find a solution. Okay, so this is, I don't want to go into this second bigger way. So approach and way forward for EC is we have to have a solution oriented approach for the EC, and prioritization has to be done along with assessment, and integration of human health and ecological risks has to be considered, and the integration of legacy, present use, and future chemicals which we can anticipate has to be listed. And then we have to have a proper modeling system, chemical analysis system, and effect-based tools has to be developed for the monitoring of EC. And then, of course, identification of EC and transformation by products. Priority has to be based on the health effects, and you also have to have understand the knowledge gap what we have with respect to the prioritized chemicals. Okay, so on top, good funding is required to do all the research. Thank you. Hello.
Thank you very much, sir. The session is now open for queries.
Bhuvaneshwari, if there is any question from the participant, allow them to directly interact with the speaker.
Yes, sir.
If there is no questions, I want to interact with the speaker.
Yes, sir. There are currently no questions on the chat box. I think you can proceed.
Is it Vinith is there?
Yes, I am here, sir.
Actually, I expected you to ask some questions with sir, because most of the topic is related to your project work.
Sir.
Okay, on behalf of Center for Ocean Research, I'm asking this to Selva Kumar sir.
Yes, sir.
Actually, the thing is, first of all, I am really happy for the elaborated manner of recording the emerging contaminant and the way how to do the analysis. Actually, this is the agenda normally we used to have here in our EACC project, particularly Mr. Vinith Kumar, who is working as one of the scholar in that project, and Dr. Karthik also being the PE in that. So we have that debate always how to extract those contaminants from these sediments and water. Hope this entire presentation give a very beautiful way of picturization where we can come with a solution. And actually, I have few questions. I ask one by one to Selva.
Please.
First thing is these emerging contaminant, now the entire focus is on about the groundwater and particularly about the drinking water. As you said, it starts from the contamination ground, and it from the land runoff, it reaches the river as well as the groundwater. So of course, in my area is the saline system. Hope there also it reaches.
Yeah.
So to my knowledge, the entire point of view, I mean, the reference, whatever available is about the detection of these kind of emerging contaminants, or as some absorption or some like that only we have come across some of the literature. The same question what I raised yesterday also, the same thing I want to ask this. Is it any device is there to completely filter out these kind of emerging contaminants, or it is any easy kit is there to identify these things?
Yeah, that is the billion-dollar question which we don't have an answer. Basically, there is no particular matrix. It's all like, for example, you take silica gel. Silica gel can be used for a wide range of matrix. Okay, so we don't have a chemically but specific matrices which can be used to extract it from the samples. But people are working on functionalization of the existing matrix to improve the binding of the EC to the existing separation matrices. Okay, so like what we did for arsenic, we did more of ion modification onto the top because iron has more affinity towards arsenic. Sorry, arsenic has more affinity towards iron. So like that, people have started working on the functionalization of the matrices for this one. Okay, so as such, one single matrix for EC is not available, to my knowledge at least. Yeah.
And one more thing, this nano extraction, actually, this is not a question. I want a little bit elaboration on those processes, the extraction of this nano contamination.
So, so basically, after synthesis of nanoparticles in our lab and utilizing it for some studies, how do we dispose it? So we don't have a concentration methodology for nanomaterials separately. Like for example, if you are using a biozide chemical, you have a biozide bin where you will put it, right? So we don't have such such things for nanomaterials. We are using chemical, what to say? We are using the same existing separation techniques for the used nanomaterials. So now how to concentrate them and how to prevent them to enter into the liquid system, into the water system. That itself is a big study. Okay, because as they are in nanometer level, our filtration, existing micron level filtration systems fail to stop them. Okay, so therefore we need to have some continuous matrix. Now what we are looking at into is the use of nanofiber system, assembled nanofiber system along with the printed structures to prevent nanoparticle entry into the water. Okay, so that we are working on it. How to use the nanofiber system basically to prevent it. So that is one way by which this solid nanometer level ECs can be prevented to enter into the water. Did I answer, Inba?
Yeah. The another question is, you said the entire EC is almost available in nanograms or in a picogram level.
Yeah.
So if it is transferred to the other tropics, maybe a primary producer or secondary any level.
Yeah.
So though it is nano or picogram level to detect in the water as well as in sediment, how we can do the monitoring in the tropical level organisms?
Okay, you have to do the monitoring based on this chart. Basically, you have to find how much of the EC which is there in the water is lipophilic in nature. Okay, and how much is non-volatile. These two things, lipophilicity and non-volatility, is going to determine the bioaccumulation into the tropics. Okay, so if it is going to be volatile, it's not going to be more into the biological system. But if it's going to be lipophilic, definitely it's going to get accumulated into the biological system. So the tropical accumulation of an EC, that means any ecosystem, primary producers or the consumers, wherever you are going to determine, you have to see which are the ECs which are likely to be more accumulated because of the lipophilicity. Okay, so the compounds, for example, if it is a polyaromatic carbon, it's going to be more lipophilic. Sorry, for example, this ones, the zoles, phenols, they are going to be more lipophilic. So therefore, you have to find basically how they get accumulated into the biological system and what is the possibility. We have to understand that. That's why understanding the physical and chemical property of the EC is the primary importance. We have to give primary importance to that before evaluating the system in which we are going to screen it. Okay, so we have to literally understand what is the chemical property of it. Yeah,
that's what I have the confusion, because the detection of this EC which is available in a nanopicogram level, which is almost depends upon the instrumentation.
yes.
Then people actually, on behalf of the students only, I'm asking this. So if there's people, some of the people, they are doing their research on the primary producers, they are exposing those animals onto those PC. So that's what maybe your answer will be a good solution for those people.
Yeah, just 2-3 points I will quickly mention on that. Like, just see the if I think you will have a focus on which chemical you are going to target, right? That at least we will know, right? In a research platform, just see the lipophilic nature of this one, and if it is going to be picogram level, then what you have to do is extract it, re-extract it, and then concentrate it. Okay, before going for concentration, you have to keep extracting it to the maximum with the same solvent system until it gets a little bit saturated, and then you have to go for the cleanup. Okay, after cleanup, you have to go for the analysis. So this is a direct link. Okay, so accumulation and concentrating the EC is more important before analysis. Even if it is picogram level per kg. So anyhow, if you are going to take a biomass, then you are going to talk in terms of grams, right? So per gram, how many picograms? So therefore, volume of sample. That's what in one of my slides I told you about the volume of sample required for extraction. That varies from one system to the other system. For biologicals, especially for the primary producer, what you're talking, these are going to be accumulated in very few picograms. Therefore, you have to take a huge volume and put an extraction system for that. Okay, so that is really a time-taking process.
Okay, Bhuvaneshwari, the other questions which is
yes, sir. There are a few questions. A question from Nirmal Renuka. Can you highlight on the pharmaceutical byproducts or metabolites and their identifications?
Okay, the pharmaceutical is an interesting EC, because, for example, you use paracetamol. What happens to the paracetamol? after degradation, it will be getting degraded inside the body. But no one is going for the quantification of them in the water. Through urine, it goes into the water. So every biological compound which is being consumed as a pharma or used as a pharmaceutical is going to give some byproducts. And you have to clearly understand what is going to be what chemical change which is going to happen from the source to the byproduct. So that needs some understanding. For example, if you are going to take one chemical and see how it get oxidizes or reduced in environmental condition, that's going to give some clues on what is going to be the byproduct. Okay, so where you will know the structure of the EC. If you know the structure of the EC, it's going to be easy. If you don't know the structure, it's a bit complex. So it's basically screening, and a lot of efforts goes into the screening part of it. Okay, so that's how you have to look into the pharma compounds. Not only pharma compounds, all the ECs. You have to take the same approach.
Bhuvaneshwari, if
Thank you.
Soumya is there, ask her to interact with us.
Yes, sir.
Soumya, are you there?
Yes, sir. I'm here, sir.
Yeah, please.
Is there any chance of losing EC in the samples while performing cleanup? Okay, the cleanup is basically the cleanup process should not actually interact with the EC compound. Okay, and if it is going to interact with the losing compound, then that's not a right cleanup method. Okay, so that's where you concentrate it, and then we always cross check the concentration of the EC before and after cleanup, just to understand whether it is interacting with the cleanup media or the solvent system. Okay. Pre-treatment, yes, we have. There are some pre-treatment models, like as I told you, when you take a soil system, even before extracting it, you use ultrasonication or microwave, which actually transforms the EC, right? So therefore, EC is removed from the solid matrix and comes to the liquid matrix, and then you start pulling it up, cleaning it up, and then analyze. So cleanup is a very important step, and I told you the cleanup methodology for GCMS and LCMS. Okay, so LCMS, it should not have any proteins. You make sure that you literally have to do a protein analysis before injecting the sample into the LCMS. Similarly, for this one, sulfur analysis is very much important for gas for GCMS. Okay, so these two are and the lipid content. You have to see whether this is there or not. So if your sample after cleanup is having both sulfur and lipid, then you are most probably the cleanup method which you have selected is not correct.
Okay, sir, I have one more question.
Yes.
See, whenever we screen for an environmental contaminant in the water,
Yes.
so if you go for a river sampling or a sea sampling, is there any chance of the genomic and the community genomics affecting the water, contaminant levels or contaminant state? For example, I'm asking that the transformation of particular metal, it could be in an oxide form or it could be in any other form. Does the community of existing bacteria or microorganisms in that particular environment play a role on this?
Definitely it plays a role. We have worked with uranium samples where we have isolated the native bacteria from the uranium ore samples, which was collected from the almost 400 meters down the earth, okay, by ONGC people. So we collected the samples from ONGC and we analyzed the microbial interaction with them, with the uranium present in those samples. And we have also studied how the uranium interacts with those samples. The samples, the organisms which are surviving in those conditions definitely have a resistant pattern. Okay, the resistant pattern can be based on an accumulation. Okay, for example, we have one of the stain which we work is on baseless ligeriformis. The ligeriformis, what it does is it has a surface layer protein on top of it, which basically nucleates the uranium present in the environment. Okay, that means, for example, you have 1 microgram of uranium in 1 gram of soil. Okay, and if the organism is going to be there in that gram, what happens is this 1 gram of uranium will not be reflected in the analysis part, because the organism crystallizes the uranium on surface and keeps it on top of it. therefore you have to literally extract everything from the organism also to be more precise on quantification side. Yes, the answer is yes. Definitely the microbial ecosystem in the particular EC environment is going to have a say on the transformation as well as the quantification, both.
So if we are going to quantify any particular environmental site for any environmental contaminant, say a metal or a metal oxide,
Yeah.
so is it advisable to extract the environmental contaminant from the microbial colonies also from that particular environment, or just the sample alone will be enough?
No, you see, the microorganism is also going to be in the same sample. You are not going to separate the microorganism out of the sample. Okay. So any extraction protocol, what you are going to put inside is going to extract the metals also from the microorganism surface. But there is a but case here, because if the EC enters the microorganism, it's not on the surface.
That is where my question comes in.
Yeah. If it is taken up, then probably you have to go for a microbial extraction process and then see how much it is there. Okay, where you have to lyse the sample, or you can use some methods which will in situ lyse the bacteria inside the soil or the water sample. Okay, for example, you can rupture the microbial cells inside the soil sample before starting the extraction process. Therefore, whatever the contents inside the cells are there, it will also be leached out. Okay, therefore it will improve the effectiveness of the extraction process. So that's where, if you see the last but one slide, I have asked people, there are some literatures on using ultrasonication as one of the modality to increase the extraction of EC. Why they have used ultrasonication? Ultrasonication is that because it can literally disrupt the cell membranes. Okay. So therefore, biological entities which are present inside, whatever has been accumulated will also be exposed to the solvent which is used for the extraction process. Okay. So therefore, that's why it has to be, everything has to be looked into when you suspect something like this. Okay. And you have to use appropriate techniques to enhance the leachability of or the extractability of the EC from the water or soil sample.
Okay, sir. Thank you, sir.
Actually, here I just want to say, maybe I may be wrong for Soumya's question.
Yes.
So maybe she is asking about what is the accumulation in the microorganism.
Yes, sir. Yes.
So it is a separate science. So those definitely. Things we have to deal in a separate way. The contamination is a separate thing.
If you want to study how much the organism accumulates, definitely you have to isolate the organism separately and expose it separately to the EC, and that study is a different study, as Inba mentioned. But if you want to see the in situ presence of microorganism and its interaction within the sample without taking it out or culturing it, then definitely you have to lyse the sample.
Okay, sir. Understood, sir. Thank you, sir.
So you have another question from Nirmal Renuka.
Yes.
Can you suggest on the recent methods for the quantification of nanoplastics in the environmental samples and their accuracy?
Okay, the nanoplastics, mostly the methodology used is Gcms mostly, because most of the compounds are a little bit more volatile and more of hydrophobicity is there. And Gcms is widely used for plastic. But before that, screening of plastic is very much important. Okay, so most of us we fail in the screening part of the microplastics. So you give more importance to the microplastic screening and on to the concentration of the microplastics. Okay, and then what are the byproducts from? And byproducts has to be understood properly. I think Paromita gave a very good details about the microplastic estimation protocols. Please follow that.
So another question from Pankaj Srivatsava.
Yeah.
Which is the best method for metabolite identification in sediment samples with GCMS and best solvent used?
Okay, metabolite identification. Sorry, I have not worked on this, so therefore it's not right to comment on that. But I have very less understanding on the metabolite part. I have not worked on that.
Any other question, Bhuvaneshwari?
No, sir, that's it.
Okay.
Okay.
Thank you, sir. Thank you very much. It was a very constructive and informative session on emerging contaminants. Especially you started right from what they are till their challenges, the concentrations and the methodologies, various methodologies which are used for extraction. And also you spoke about the solutions and the approaches and way forward. Thank you so much, sir. It was very useful.
Thank you, guys.
And thank you, Selva.
Thank you, sir.
Thank you.
Thanks for the interactive session. Actually, even the entire thing we are also working, and most of the our scholars themselves they have had the queries with you. Thanks a lot. And long time pending is there. We have to meet soon. Okay.
Definitely.
Sir, Selva sir, I'm having one final curious question. Sir, have you worked any bio-based nano adsorbent for treating water?
Okay, a lot. Basically, I have worked a lot. Okay, to nutshell, we have what we have did is we have extracted microbial proteins which have higher affinity towards some compounds. This work is especially on the uranium side. So what we did is we extracted the microbial proteins from the bacteria and we concentrated it, quantified it, and then we surface functionalized them onto the nanomaterials. Okay. So what we did is that process is called a self-assembly. So some proteins have the property to self-assemble onto any matrices. S-layer protein of microorganism is one such protein which has the capacity to assemble onto matrix. Okay. So we can use that process, and we have increased the extraction of uranium from water by 30% when compared to normal nanomaterials. So that means when you integrate a biological compound to a nanomaterial, we can literally increase the extractability of metals, especially like uranium and thorium from water. Yeah.
Any plant-based?
No, ma'am. Most of my studies are all on microorganisms.
Okay.
Being microbiologist, I don't go into other areas. Yeah.
And one more thing, actually, Selva, he's handling a project from mining. So Ministry of Mining, he's handling a project. So I think Pakilashmi ma'am now have a clue. She can contact him, and there is now there is a call for a mining.
Yeah.
So that will work out, man.
15th April is the last date.
15th April is the last date.
Yeah,
who are being here and they want to work on that, they can also work on that proposal.
Please feel free to contact. Yeah.
Yes, sure, sir. Sure.
Yeah.
Thank you, Selva.
Thank you. Sorry, Gagan sir, I have taken your time.
No. It's not today. It's not his.
His lecture is not today.
26. It's on 26.
Sorry.
No
Okay. Fine.
Okay.
Thank you.
Thank you.
Is there any other questions? Okay.
Yes, sir. I guess you have one last question, sir. It's on the chat box from Ria. While selecting a specific treatment process that removes some of the target emerging contaminants in water, how important it is in analyzing the mineralization efficiency after a specific treatment, especially when we don't know the reaction pathway or potential byproduct formed?
Very interesting question, and in fact, very important question too. And because the TOC is going to give a clue on what is the level of contaminants inside it. Okay, so whether the extractability is efficient or not, that's going to be every stage. If you just take TOC into account, it's going to give us some clue on what is the efficiency of the method which we are going to use. Okay, so TOC definitely is going to be an important factor for the treatment process, not only in treatment process but also in the screening process.
Thank you, sir. And also doing a mole balance might help, like, in different stages, like to know how many cycles to go.
See, since this area is quite new and lot of data is not there, that means a lot of research is required and very less data is there. We have to literally optimize every step of EC before we start working. We have to think a lot, we have to plan a lot before addressing the EC in water because of derivatization, not only because of that accumulation process and many things. Okay, We say that it will be 100 microgram, but it will be either more or less because of the derivatization. Okay.
Yes, definitely.
Yeah. Thank
And
you.
byproducts could have toxicity different from the parents. So that is
I
also
think
a by.
it's a field by itself, basically.
Yeah, definitely. Yeah.
It
So
has to be
byproduct
totally.
analysis is equally important. Like we don't have methods for all byproducts. Like even when we use external standards, we cannot really find some standards for these. Some of the like when we do chemical degradation, especially certain the ring breaking, we don't have structures that we can have like external standards to quantify. So that can be really important there.
That is the biggest challenge. We don't have standards.
Yeah.
And we don't understand how it breaks. That is again a problem. That means where it is getting cleaved, on which factors are influencing the cleavage. Nothing is understood properly. And, a lot of insight is required on EC, basically.
Yes. Thank you, sir.
Thank you, Dr. Selva.
Thank you, sir.
Thank you, sir.
Thank you so much.
Bhuvaneshwari, you can go ahead with the announcement.
Yes, sir. Dear participants, kindly note the new Zoom link for tomorrow's technical session. That is on 24th of March 2022 from 10:15 a.m. We'll be having with us Dr. R. Saravanan, Associate Professor, Center for Water Resources, Anna University. He'll be delivering his lecture on sustainable water management, critical issues, and strategic options. I hope you all have noted down the link.
So the same link will be emailed to you?
Yes.
Also. Yeah. Thank you.
For the same link for all 3 days, remaining days.
Yes, sir. Yeah, yes, ma'am. Yes, ma'am. This is the new link, and the same link will be used for all the 3 days, ma'am.
Okay, thank you all.
Thank you, Selva. Thank you, Selva. Thank you.
Thank you.
Thank you.
Thank you
Thank you
all.
all the participants. Thank you.
Bye.